Okay, so hi everyone, my name is Matthew. I am a final year PhD student with the National University of Singapore and Hebrew University of Jerusalem. So I've actually just submitted my thesis a few months ago, and right now I'm just hoping for the best. So the work that we have done these past 4 years or so revolves very much around a subset of diseases known as chronic inflammatory diseases, or CIDs for short. For example, those of the lung, which I actually focus more on, such as asthma and chronic obstructive pulmonary disease. Now, these diseases afflict millions of individuals worldwide. They're also very prominent causes of mortality amongst people, and at the same time, they pose many severe burdens in terms of quality of life as well as to the economies of the various countries. Now, the thing about these diseases is that although we have studied them for quite a while, I would say, we still don't actually know what causes them. We have no idea what causes a disease to develop in one person but not another. For example, we have heavy smokers who live the rest of their lives without actually suffering from any of them. And yet we also have cases where people who have not touched a single cigarette in their entire lives before end up developing COPD. So the work that we have done throughout the course of this PhD is actually attempting to look at the potential underlying molecular mechanisms that may lead up to the development of such diseases. And we focus on a particular pathway that seems to be upregulated and highly active across all these diseases. And yet, until this point, this pathway has seen decades of effort and work basically fail to translate into a viable therapeutic for the clinic. In order to really explore why this is so, we have actually created and designed a novel transgenic mouse system that allows us to, for the first time ever, trigger this pathway alone, on its own, and free from any other, I would say, confounding factors, in order to truly elucidate and distinguish what happens when we activate this particular pathway. So I will now share more and describe kind of the design that we used in order to develop this transgenic mouse system. So our transgene contains two expression cassettes within them, the RTTA expression cassette, as well as another cassette downstream for expression of our active P38 variant. So the first cassette on the left, as we see in the schematic here, is our RTTA expression cassette, which codes for the RTTA-M2 transcription factor under the control of a constitutively active KEX promoter. However, what we have done here is actually to add a stop termination sequence in between the promoter and the RTTA gene, which means that it cannot be transcribed under normal circumstances. However, this stop sequence is also flanked by LOXP sites, which allows it to be excised in the presence of the enzyme Cray recombinase. As such, RTTA-M2 will only be transcribed in cells that express Cray recombinase already. Downstream of the RTTA transcription cassette, we have what we just term as the active P38 expression cassette, which actually contains the gene encoding our active variant. So in this case, the gene here is actually controlled by a TET-on promoter system that requires RTTA-M2 in order to drive transcription. However, for RTTA-M2 to actually bind to the TET-on promoter, it needs to first form a complex with a tetracycline molecule, for example, doxycycline. This RTTA-DOX complex can then bind to the TET-on promoter in order to drive downstream gene expression. So what we have here in our transgenic mice is a combination of the two cassettes, and it actually allows us to express these active P38 molecules under tight spatial and temporal control. It can only be expressed in cells that already express Cray recombinase, and only in the presence of tetracyclines such as doxycycline. Another advantage that it actually has over all other current methods of studying P38 is that this is the only way in which we can activate P38 alpha alone and prevent the unwanted concomitant activation of other major signal transduction pathways. Thus far, we have actually validated this concept in a full body expression model of our transgenic mice, where we actually express the active variant under the control of the actin promoter, which would technically and theoretically mean that it is being expressed throughout all cells of the body. We have validated that the system works as it was intended to, and that we actually have very nice and very tight spatial and temporal control over expression of the active variant with very little, or I would say negligible, leaky expression being observed even for time periods of over a year. And we have also brought it one step further, in which we have expressed this transgene of ours under the control of tissue or cell-specific promoters, for example, the albumin promoter in the liver or the surfactant protein C promoter. So the work that we have done during the course of this PhD with the various transgenic mouse models that we have actually shown that our concept works as it was intended to. We were able to very tightly control both the where and the when our active P38 variant could be expressed, and this ensured that for the first time ever in a in vivo living organism, we were able to activate P38 alone, free from the confounding effects of other major signal transduction pathways. Interestingly, some of the findings that actually came out of this, they actually seem to go against what would be commonly expected. But in order to actually find out more, I would suggest that you check out the publications that have come out from our lab, where we have actually shown that P38 alone could actually be sufficient to trigger certain pathologies. And not only that, it appears to be doing so not necessarily by the upregulation of its downstream signaling components, as is commonly assumed, but rather it seems to be causing the downregulation and the degradation of some of these components instead in the long term. How this actually leads to disease and all remains to be seen, but hopefully as we continue to work on these transgenic mouse models, they will help to shed light on the causes behind these chronic conditions and perhaps one day lead to the development of more efficacious and definitive therapeutics and cures for them. Thank you.